Namaste, and welcome back to Typical Monk. We saw the structure and the copying of DNA. Now we see how DNA writes its message into RNA, that is, transcription. It is the first step of gene expression, one idea at a time. Here is today's path. 5 stops. First, what transcription is. Second, the transcription unit. Third, transcription in prokaryotes. Fourth, transcription in eukaryotes. And fifth, split genes and RNA processing. First, what transcription is. Transcription copies a gene from DNA into RNA. Only one strand is copied. The template strand read 3' to 5'. The other strand, the coding strand, is not copied. It simply matches the RNA. RNA polymerase builds the RNA in the 5' to 3' direction. Now, the region that is transcribed, the transcription unit. A transcription unit has 3 parts: a promoter, the structural gene, and a terminator. The promoter lies at the 5' end and tells RNA polymerase where to start. At the 3' end, the terminator marks where to stop. Now, in prokaryotes, a single R on A polymerase makes all the RNA. A sigma factor helps initiation, and a rho factor helps termination. With no nucleus, transcription and translation can happen together, that is coupled. Now, in eukaryotes, here there are 3 RNA polymerases. Look at this table. RNA polymerase 1 makes urea, RNA polymerase 2 makes MISNA, which first forms as nRNA. RNA polymerase 3 makes TANA, 5 SARNA, and small NAS. In eukaryotes, transcription and translation are separate, one in the nucleus, one in the cytoplasm. Now, a special feature of eukaryotic genes, they are split. Look at this picture. Eukaryotic genes are split. Exons, the coding parts, in green, and introns, the non-coding parts, in gray. The first transcript is RNA. It still contains the introns. Splicing removes the introns and joins the exons. Then, a 5' cap and a 3' poly a tail are added, and the mature mini is ready. Now, the high-yield pointers, the facts that NEET turns into questions. 1. Only the template strand, that is 3' to 5', is transcribed. RNA is built 5' to 3'. 2. The unit is promoter, structural gene, and terminator. The promoter is at the 5' end. 3. In prokaryotes, 1 RNA polymerase, sigma means initiation. 4. In eukaryotes, polymerase 1 makes RNA, 2 makes MANA, that is an RNA, 3 makes CHANA. 5. Processing, capping, tailing, splicing, introns out, exons expressed. Now the first drill, assertion and reason. Each time, ask, is it true? Is R true? And does R explain A? First, A in eukaryotes. RNA must be processed before it can work. RNA contains introns that must be removed. Both are true, and R explains A. So the answer is A. Second, the coding strand is used as the template in transcription. RNA polymerase reads the template 3' to 5'. Here A is false, but R is true, so the answer is D. Third, introns do not appear in mature MINA. R splicing removes the introns. Both are true, and R explains A. So the answer is A. Now, the second drill, match the column. Make the correct pairing. RNA polymerase 1, RNA. RNA polymerase 2, MISNA, that is nRNA. Sigma factor, initiation. And coding regions, exons. Now the third drill, how many of these statements are correct? Check them one by one. First, exons are the coding, expressed sequences. This is correct. Second, in eukaryotes, a single RA RNA polymerase makes all RNA. This is wrong. Third, the promoter lies at the 5' end of the transcription unit. This is correct. So the correct statements, 2, that is, the first and the third. Now let's clear the mistakes students fall for. Myth, introns are the coding parts of a gene. Truth, exons code and are expressed, introns are removed. Myth, both T and A strands are transcribed into RNA. Truth, only the template strand is. The coding strand is not. Now test yourself. Answer each one before it is revealed. Answer first. That is how you build strong NEET recall. Question 1. Which strand is transcribed? Answer. The template, that is 3' to 5'. Question 2. Which RNA polymerase makes messenger? RNA polymerase 2. Question 3. The coding parts of a split gene. Exons. Question 4. The prokaryotic initiation factor. Sigma. Question 5. Removing introns is called splicing. Let's gather it all in one breath. Transcription, DNA to RNA. Only the template strand copied. RNA made 5' to 3'. The unit, promoter, gene, terminator. Prokaryotes, 1 RNA polymerase, sigma to start, rho to stop. Eukaryotes, polymerase 1, 2, 3, make RNA, marna, tarna. Split genes, exons and introns. Processing is cap, tail, splice. In the next class, the genetic code and translation. See you there. You have understood how the cell writes its message. Be proud. One idea at a time with a calm mind, and you will crack NEET. This is typical monk. Every class, always free.